Hello team, and welcome to our second screencast from chapter 17. In this screencast, we're going to cover sections 17.3 and 17.4. In section 17.3, we're going to talk about the chemical hazards that we face in our environment. The word toxic means harmful, and toxic chemicals fall into different categories based on the type of harm that they do. Carcinogens are chemicals or other agents that can cause or promote cancer. Mutagens are chemicals or other agents that can cause mutation or increase the frequency of them. The symbol for cancer hazards can be seen here in the image. Teratogens are chemicals or other agents that can cause harm or birth defects in developing embryos and fetuses. The diagram here shows where teratogens commonly act. It also shows the potential problems that can arise from teratogen exposure. Polychlorinated biphenyls, or PCBs, are good examples of toxic chemicals. These chlorine-containing compounds are stable, non-flammable liquids at room temperature, and as a result, they had many industrial and other uses. Unfortunately, PCBs are what we call persistent organic pollutants, or POPs. POPs break down slowly, and as a result, they linger and stay in the environment for long periods of time. PCBs are known teratogens and probable carcinogens. PCBs are also fat-soluble, which means they bioaccumulate in the tissues of organisms and biomagnify in food chains and food webs. PCBs are banned, but because of their persistence, they're still found everywhere in the environment. This diagram shows the many uses of PCBs, everything from adhesives to casting wax. This diagram shows how PCBs can go from the environment into the tissues of organisms and up the food chain into other organisms. This diagram shows how the biomagnification of PCBs occurs. At the bottom of the food chain, phytoplankton absorb PCBs from their environment. As organisms eat one another, the concentration of PCBs increases until it reaches its maximum concentration in the top predator, which is the orca whale here. Some chemicals, both natural and synthetic, can weaken and harm the immune and nervous systems. Chemicals that affect the immune system weaken it, affecting its ability to fight off infection, illness, and disease. Chemicals that affect the nervous system are called neurotoxins. Examples of neurotoxins are PCBs, arsenic, lead, some pesticides, and methylmercury. The diagram here shows how mercury can affect the development of a fetus. There are two ways in which we can deal with toxic chemicals in the environment. First, we can prevent them from making their way into the environment. Secondly, we can control people's exposure to them. This table shows how we can both prevent and control mercury pollution. Other chemicals can affect our endocrine systems. The endocrine system is the system of glands that release hormones to regulate bodily systems and control sexual reproduction, growth, development, learning, and behavior. Examples of organs in the endocrine system include the pancreas, the thyroid gland, the testes, and the ovaries. Hormonally active agents, or HAAs, are a type of toxic chemical that affect the endocrine system. They act by having similar shapes and binding to hormone receptors. They can have one of two effects. Hormone mimics act like or copy other organisms. For example, BPA found in plastics can mimic or copy estrogen. Hormone blockers stop or block other hormones from working. For example, some pesticides can block testosterone. In recent years, there's been a movement away from BPA in plastics because of its ability to mimic estrogen. Atrazine is a pesticide and blocks the action of testosterone. A research study found that atrazine prevented 75% of male frogs from reproducing and caused 10% of the male frogs to become female. This fueled conspiracy theories peddled by people like Alex Jones on the left here, saying that the government was trying to taint the water supply and affect people's sexual development and sexuality. In section 17.4, we'll talk about how we can evaluate and study chemical hazards. Toxicology is the study of the harmful effects of chemical substances. The toxicity of a chemical can depend on a number of different things, including dose, age, genetic makeup, individual sensitivity, which causes a condition called multiple chemical sensitivity, or MCS, solubility in water or bodily tissues, persistence in the environment, and biomagnification. Exposure to chemicals in the environment can have immediate or rapid effects. These are called acute effects. Or they can have permanent or long-lasting effects. These are called chronic effects. Estimating chemical exposure and predicting the effects of that exposure on people is very difficult to do and includes a lot of different factors. Scientific measurements and modeling by toxicologists attempts to do this. Toxic chemicals affect children disproportionately. A toxicological analysis of umbilical cord blood from newborns found 180 chemicals known to cause cancer in human and other animals. Infants and children are more susceptible because they have less developed immune systems and body detoxification processes. We're especially concerned because there seems to be a link between fetal exposure to chemicals and increased risk of autism, asthma, and learning disorders. Studying toxicity on human beings can be ethically difficult. As a result, toxicologists use mice and rats. These animals have the added advantage of being small and reproducing rapidly. Toxicologists will give these animals different doses of a chemical and measure the response. This produces what we call a dose-response curve. A dose-response curve can be used to determine the median lethal dose, or LD50. This is the amount of a chemical that will kill 50% of a test sample. We can also use the dose-response curve to figure out the threshold dose. This is the amount of a chemical in which we first see a specified and measurable effect. This is an example of a dose-response curve. It measures the lethality of a chemical at different doses. You can see the median lethal dose, or LD50 here, somewhere between 6 and 8 hypothetical units. You can also see the threshold dose, which is somewhere between 2 and 4 hypothetical units. Toxicologists use two different dose-response curve models in order to estimate the toxicity of various chemicals. The non-threshold dose-response model has non-linear responses even at low doses. The threshold dose-response model has no response until a threshold is met. After the threshold dose, there's a linear response. The non-threshold dose-response model is shown here on the left with the solid red line. The threshold dose-response model is shown on the right side with the dashed green line. More and more frequently, people are raising objections to using animals in lab research. Fortunately, we can use more humane methods to study toxicity. Toxicologists are using computer simulations, tissue culture, individual animal cells, and chicken eggs in which the membrane inside is exposed to different chemicals. Moving forward, we also need to study the effects of mixtures of potentially toxic chemicals. You may have heard of drug interactions between alcohol, medicines, and other drugs. There are other ways in which we can estimate the harmful effects of chemicals. Epidemiology is the study of the patterns, causes, and effects of health and disease conditions in populations. For example, the map on this slide shows the results of an epidemiological study of the effects of lead in the water supply in Flint, Michigan. The most mild effects are in green, the most severe effects are in purple. Epidemiological studies do have limitations. First, sometimes not enough people are tested and sample sizes are small. Second, sometimes not enough time is permitted to see the effects of chemicals that might not appear until years or decades later. Third, sometimes it's difficult to establish a strong link between health effects and a chemical. And finally, epidemiological studies are not good for hazards that we know little or nothing about. This map shows the results of an epidemiological study that traced the movement of the Zika virus from Africa in 1947 to South and Central America between 2014 and 2016. Is low exposure to chemicals harmful to us? We don't know because there's insufficient data for most chemicals. Unfortunately, we're exposed to many toxic chemicals in low amounts, which are also called trace amounts. This diagram shows how we're exposed to trace amounts of toxic chemicals throughout our homes. There are severe limitations in our ability to estimate the toxicity levels and risk of different chemicals. Only about 2% of 100,000 chemicals that we can be potentially exposed to have been adequately tested. On top of that, 99.5% of chemicals used in the United States are not supervised by government. One of the best ways to deal with chemicals in the environment is to use the precautionary principle. We should assume that any new chemical product is harmful until it is proven to be safe. We should also remove any chemical or technology that appears to cause any significant harm. Governments around the world are trying to manage the risk caused by toxic chemicals. In 2000, a global treaty was signed that banned or phased out the use of 12 persistent organic pollutants, or POPs. Because there were 12 of them, they were known as the Dirty Dozen, and they're listed on the left here. In 2007, the European Union created the REACH program that shifted the responsibility of chemical safety to the companies that created or used them. Individual companies are also trying to minimize chemical pollution and exposure. Between 1975 and 2008, the 3M company, most famous for Post-it notes and Scotch tape, prevented more than 1.5 million tons of pollutants from reaching the environment. They did this through an employee reward program that rewarded people for finding ways of minimizing chemical waste.